السلام عليكم. إن شاء الله our next lecture بعد ما أخدنا the structure and characters of amino acids which are the building blocks of the protein. تعالوا نشوف البروتين conformation and structure وازاي بيحصل له folding to form functional protein. البروتين بقى اللي بيعمل لنا الفانكشن in our body. الامينو acids are the building block. لازم تبقى connected مع بعض علشان تعمل لنا different structures and conformation of protein. There are 4 levels of the structural organization of the protein. احنا هندرس ال4 levels. اول حاجة انا عايزاك تعرفها في كل ليفل هو it consists of what، البوند بتاعته ايه. البوند دي it's a chemical bond تعمل stabilization of this structure. ف each level have a specific bond that stabilize it. أول level من ال 4 levels of the structure of the protein primary level which is determined by the number and sequence of amino acid in the protein which different from one protein to another. Each protein have a specific sequence and number of amino acids. Second level called secondary structure which is the conformation of the polypeptide chain, and this is formed by twisting or folding. It occurs when amino acid linked by hydrogen bonds to form either alpha helix or beta pleated sheets. كل ده ان شاء الله هنشرحه بالتفصيل. يعني احنا الاول we will summarize the levels of structure of the protein. Each level we will explain in details. Tertiary structure, it's the third level. The three-dimensional arrangement of the protein structure and formed when the alpha helix or the beta sheets are held together by weak interactions or weak bonds. Each level has specific bonds زي ما قلنا. The fourth level called quaternary structure, and this occurs when the protein oligomers. Oligo means a number of subunits from 3 to 9. يبقى some number of 3 to 9 subunits form the functional protein called oligomers, consist of more than one polypeptide chain, and they aggregate together to form the functional protein. ده هو اسمه the quaternary structure اللي هناخد عليه examples في our next 2 lectures في الكولاجين as fibrous protein والهيموجلوبين والمايوجلوبين as globular protein. This is the 4 ordered structure of the protein. Let's see the figures to explain and to understand the levels of the protein. This is the primary structure, which is the sequence of amino acid by specific sequence and number, and they are connected by what's called the peptide bond. This is the bond that stabilizes the primary structure of the protein. This is the primary structure. Next, secondary structure. Secondary structure when the polypeptide chain is twisted. This is twists or folded. This is called alpha helix or beta pleated sheets. And this is stabilized by hydrogen bonds. What called hydrogen bonds. يبقى ال secondary structure when the number and sequence of amino acid is twisted in the form of alpha helix or beta pleated sheets and stabilized by hydrogen bonds. And this is tertiary structure. It's the three-dimensional structure or arrangement of the protein structure and stabilized by weak interaction bonds. And this is the quaternary structure when oligomer. It's a number of subunits from 3 to 9 connected together to form the functional protein. This is the quaternary structure. Let's take each level in details. The primary structure of the protein, which means the number and sequence of amino acid in the protein. They are linked by peptide bond. Peptide bond, it's a type of the covalent amide bond. This peptide bond occurs between the carboxylic group of one amino acid and the amino group of the successive amino acid, the adjacent amino acid. It loses a molecule of water and form peptide bond. Generally, amino acids are arranged in linear structure called polypeptide chain, and each amino acid is called resid or moiety, connected by the peptide bond. At the end, each polypeptide chain will have free amino end or N terminal at the left and free carboxylic end, it's called C terminal on the right. احنا قلنا ان الconfiguration of the amino group in all amino acids it's L on the left, so ال N terminal تبقى in the left وال C terminal on the right. يبقى free groups, not connected, not entering information of the peptide bond. The amino acid sequence and number is determined by the genes on DNA. لو فاكرين translation of DNA, الجينز coding protein هي اللي determine the structure and the sequence of the amino acid and the number of amino acid in each protein or each polypeptide chain. And also it determine the physical and chemical property of the protein. What is the peptide bond? It's covalent bond formed by the amide linkage between the alpha carboxylic group of amino acid and alpha amino group of another amino acid, the successive amino acid. We remove one molecule of water and form the peptide bond. This is what called the peptide bond between the carboxylic group of one amino acid and the amino group of the adjacent or successive amino acid. It's strong bond and form strong backbone of the polypeptide chain. The polypeptide chain تبقى linear and there is a side chain of the amino acid will project outside from this backbone. A dipeptide means two amino acids connected by one peptide bond. Tripeptide means three amino acids connected by two peptide bond and so on. Okay, يعني dipeptide means two amino acid وفي one peptide bond بس. Tripeptide bond means three amino acids and two peptide bond and so on. تعالوا نشوف الفيجر. It will explain better the primary structure. This is the peptide bond. This is the first amino acid, the second amino acid. They are connected by removal of one molecule of water to form the peptide bond. between the carboxylic group of amino acid and the amino group of the next or the successive amino acid, forming the peptide bond, and then this will form the linear polypeptide chain. This is the linear polypeptide chain connected by peptide bonds between the successive amino acids, and this is the moiety or amino acid residues. At the end it will have N terminal free amino group on the left and C terminal free on the right. This is the linear polypeptide chain, which is the primary structure of the protein. ده الفيرست level. Secondary structure of the protein, which is the second level, it's the three-dimensional conformation of the polypeptide chain by twisting or folding, and this is called the secondary structure of the protein. It have many kinds. alpha helix شفنا له figure قبل كده وهنشوف تاني. beta pleated sheets, the stretched state, the loop region, and the beta bend or the beta turn. They are all secondary structures or twisting of the polypeptide chain. Let's see the difference between different kind of the secondary structure of the protein. The alpha helix is the most common stable conformation which have the lowest energy in the secondary structure, stabilized by this is important, by hydrogen bond of the main chain. ليه؟ لأن الألفا هيليكس just contain one polypeptide chain. Form the backbone، يبقى فيه hydrogen bond formed in a single polypeptide chain, make it twisted or folded to form the alpha helix. Hydrogen bonds occur between the carboxylic oxygen of one peptide bond and the amide nitrogen of another peptide bond، لكن ما بيكونوش successive to each other، بيكون في 3 amino acid apart أو further down in the chain. All polypeptide bonds except the first and the last peptide bond can participate in the hydrogen bond. The first and the second not participate in the hydrogen bond. Proline, hydroxyproline, glycine بيعملوا disruption of the alpha helix formation. They introduce a kink or a bend on the alpha helix. هو بيبقى linear لو الامينو اسيد برولين، هيدروكسي جلايسين بيعملوا kink، وده هيجيلنا in details في الكولاجين. Also large number of acidic amino acid, aspartate or glutamate, basic amino acid, lysine, arginine, histidine, or amino acid with a bulky side chain will disrupt the alpha helix. Alpha helix structure is abundant in myoglobin, hemoglobin, and keratin, which is a fibrous protein. We will mention it in details in next lecture. Let's see the figures. We will understand more by the figure. This is the alpha helix. It have free N terminal. It have free C terminal. It's twisted. Each turn travel a distance of 5.4 nanometer per turn, and the axial ratio it's 5.8 angstrom or nanometer per turn. This is the hydrogen bond. يبقى when I ask you what is the bond stabilizing the secondary structure of the protein, it's the hydrogen bond between C CO carboxylic group of one peptide bond and NH of another peptide bond, and there is 3 amino acid apart, not successive to each other. Look at the turn of the alpha helix. This is the bond, hydrogen bond between amino acids, not successive, 3 amino acids apart to form the alpha helix. This is another figure of the alpha helix. This is here the hydrogen bond between, this is the CO of one peptide bond and NH of another peptide bond of another amino acid, not successive, 3 amino acids apart to form the hydrogen bond. The pink one is the hydrogen. Why it's called hydrogen? Because the hydrogen enter in the formation of the bond. It's called hydrogen bond, the pink one. This is the beta sheets. You will know the beta sheets is sheets parallel to each other, and the hydrogen bond formed perpendicular to the sheets. This is the pink one, is hydrogen bond between carboxylic group of one peptide bond and NH group of another peptide bond forming hydrogen bond. This is the hydrogen bond. The beta pleated sheets, it's one type of the secondary structure. يبقى the first type is the alpha helix. We still in the secondary structure or second level of the protein structure. We talked about alpha helix. This is the second type, is the beta pleated sheets. Beta pleated sheets are stabilized also by hydrogen bonds. They are composed of 2 or more segments of fully extended polypeptide chain or 2 polypeptide chain may run in the same direction, so they are called parallel beta pleated sheets, or run opposite to each other, so they are called anti-parallel pleated sheets. Look at this figure. The first figure is the parallel beta pleated sheets. They run in the same direction. The N terminal of all of them at the left side, C terminal of all of them on the right side, they are parallel. The next figure is anti-parallel beta pleated sheets. N terminal is opposite to C terminal of the other polypeptide chain, and the opposite in the other right side. It's anti-parallel. Let's explain on the figure. In the beta pleated sheets, the hydrogen bonds has 2 types, either interchain or intrachain. What is the meaning of interchain? It's formed between the amide hydrogen of one polypeptide chain and the carboxylic oxygen of the neighboring polypeptide chain. يبقى هنا داخل 2 polypeptide chain. But the intrachain, intra means جواها, in. So a hydrogen bond formed between the amide nitrogen of one polypeptide chain and the oxygen of the same or the carboxylic group of a peptide bond in the same polypeptide chain. So they are called intra, in. Inter is between. Intra is in. Inter is between 2 polypeptide chains. Intra is inside one polypeptide chain. Let's see the figure. This is the beta pleated sheets. They are parallel or anti-parallel. Look at this. This is the N-terminal and C-terminal. So they are anti-parallel. They are anti-parallel. There is here is the peptide bond. When one carboxylic group of one peptide bond form hydrogen bond with the amide nitrogen of another peptide bond, this is called the hydrogen bond. This is intra or inter-chain hydrogen bond. It's inter between two polypeptide chain. In this figure, this is inter-chain hydrogen bond. You see with me? So here they are anti-parallel beta pleated sheets. This is the peptide bond here, peptide bond, the red one, okay? And carboxylic group of one peptide bond with amide group of another peptide bond in the other polypeptide chain will form hydrogen bond, and this is called interchain, يعني between two polypeptide chain. We understand with me? And this is called the bend or the loop. We will explain it in the next slide. This is called beta bend or beta loop. It's between the end of the anti-parallel polypeptide chains, and there is twisting in the polypeptide chain. It changes its direction. Here is it's changing in its direction, and it's formed of 4 amino acids to form the beta bend or the beta loop. We will explain it in details next slide. Here are, this is carboxylic group of one peptide bond, and this is the amide nitrogen of another peptide bond, okay, here, and this is the dotted one is the hydrogen bonding. The dot, you look at the dotted line here, the dots between the carboxylic and the amide of another peptide bond, hydrogen bonding. What is the difference between alpha helix and beta pleated sheets? Alpha helix is a coiled rod-like. Beta pleated sheets are fully extended sheet-like. Hydrogen bond on the alpha helix, they are parallel to the polypeptide backbone. They are parallel. This is the direction of the polypeptide backbone, and they are parallel. But here, the hydrogen bonding is perpendicular to the direction of the polypeptide chain. This is the polypeptide chain, and this is the direction of the hydrogen bond, perpendicular. The axial distance between the amino acid is 1.5 angstrom in the alpha helix, and in the beta pleated sheets 3.5 angstrom. The alpha helix, it's formed of just a single polypeptide chain twisted, but the beta pleated sheets can be one polypeptide chain twisted into segments, or more than one polypeptide chain. It may be 2 or 3. So alpha helix is single polypeptide chain, but beta pleated sheets can be a single or more than one polypeptide chain. Here are the beta bend or the beta turn or the hairpin turn or the reverse turn. I explained in the previous figure. It's when the polypeptide chain abruptly reverse its direction and connect the ends of adjacent anti-parallel beta strands. They are named beta turn, beta bend, hairpin, or reverse turn. There is changing in the direction of the polypeptide chain. It's abundant in the globular protein like hemoglobin, myoglobin. We will explain it in details in our next lectures. Characters: they consist of 4 successive amino acids residues. They are frequently contain proline, glycine, or both. احنا قلنا قبل كده they disrupt the alpha helix and form a kink. طب الكينك دلوقتي we need it to form the turn. So they are abundant in proline or glycine or both as they form a kink. They are stabilized by intra-chain, not intra-chain disulfide bridges and hydrogen bond. Here is another or a new bond, the disulfide bond. We will explain it in details in the tertiary and quaternary structure. Disulfide bond and hydrogen bond formed between the first amino acid and the fourth of the bond. احنا قلنا هي 4 amino acids. البيبتايد bond form الهيدروجين bond is formed between the first and the fourth. Also there is intra-chain disulfide bond. This is the beta bend or the beta turn. There is what called super secondary structure of the protein. Super secondary means that there is a specific arrangement of the secondary structure of the protein to form what called motifs. Motifs means a domain or a functional part of the protein. So when the protein molecule is arranged in the form of many types of secondary structure, يعني ال super secondary ده can contain alpha helix followed by beta pleated sheets. So we called it super secondary, and it form or even contain beta turn or loop region, يعني can contain more than one type of the secondary structure. So it's called super secondary. It form or recognized as domain or motif, and it's abundant in the globular protein. Let's see the types of the super secondary structures or motifs. They contain more than one type of the secondary structure and forming secondary structure element called motifs. The first one on the left is beta alpha beta motif. Look at this. It's beta and then a turn or a bend and alpha helix and another beta chain. The second also on the left down is the beta minder motif. Minder means zigzag like. It's twisted, means twisted. It's formed of beta pleated sheets and beta bend. And the Greek key motif on the right up on the figure. This figure is called Greek key. And also right but down of the figure, the beta barrel, it's barrel like. This is the super secondary structure which form motifs or domains.